At a depth of 8,178 meters in the Mariana Trench, a small, translucent fish drifts through water that is pressing on every square centimeter of its body with a force of more than 800 kilograms. The fish is the Mariana Hadal snailfish, Pseudoliparus soiree, and it is the deepest living fish ever recorded. Its body is pale, gelatinous, and poorly muscled. Its eyes are small but functional. Its skin is so thin that the outlines of its internal organs are visible through the body wall. It looks fragile, delicate, ephemeral, as if it could not survive in the water column above a kitchen sink, let alone at a depth that would crush a nuclear submarine. The fish is not fragile. It is one of the most precisely adapted organisms on the planet. Every molecule in its body has been modified by natural selection for the specific conditions at the bottom of the deepest trench in the ocean. Its proteins are stabilized by a chemical that no surface organism possesses at comparable concentrations. Its cell membranes are composed of lipids that maintain their fluidity at pressures that would solidify the membranes of any surface fish. Its genome has been rewritten, with genes lost that are no longer needed and genes amplified that are essential for life at the extreme. The fish is not surviving at the bottom of the trench. It is thriving, adapted to conditions so extreme that the biology needed to describe its adaptations was not understood until the 21st century. The Hadal Zone, the ocean below 6,000 meters, is the last frontier of biology on this planet. It consists of the ocean trenches, the deep, narrow V-shaped gashes in the seafloor that are formed where one tectonic plate dives beneath another. There are roughly 33 Hadal trenches worldwide, distributed primarily around the Pacific Rim, and they contain conditions that are found nowhere else on Earth. Conditions that include the highest pressures in the ocean, unique chemical environments, unique food supply dynamics, and unique patterns of isolation that have produced organisms with biology that is as strange and as specific as any on the planet. By the end of tonight, you are going to understand what makes the biology of Hadal Zone creatures so strange, what molecular adaptations allow them to function at pressures that would disable every surface organism, why there is a depth beyond which fish cannot survive, what replaces fish below that depth, and what the biology of these extreme organisms reveals about the limits of life on Earth and the potential for life on other worlds. Before we get started, if you find these mysteries as fascinating as we do, take a second to like the video or subscribe. It is a simple action, but it helps this channel reach more curious minds like yours. The Hadal Zone contains roughly 45% of the ocean's total depth range, but less than 2% of its total area, concentrated in the narrow trenches that trace the boundaries of the tectonic plates. The zone is small by area, but extreme by every other measure, and the organisms that inhabit it possess adaptations that are found nowhere else in the ocean, adaptations that are as specific to the Hadal as the adaptations of the desert are to the arid, or the adaptations of the polar are to the cold. The Hadal organisms are not merely deep-sea organisms that happen to live at the bottom of trenches. They are Hadal organisms shaped by the specific conditions of the Hadal, with biology that is distinct from the biology of the abyssal organisms that inhabit the seafloor above 6,000 meters. The distinction is not trivial. It is molecular, genomic, metabolic, and ecological. The Hadal organisms are different from the abyssal organisms at every level of biological organization, from the molecules that compose their bodies to the ecosystems that they inhabit. The strangeness of the Hadal biology is a consequence of the extremity of the Hadal conditions, conditions that push the molecular machinery of life to its absolute limits, and that produce organisms whose biology is as alien to the surface biologist as the biology of any organism in the solar system. The Hadal Zone was, until recently, assumed to be essentially lifeless. The pressures were considered too extreme, the food too scarce, and the isolation too complete for complex biological communities to persist. The assumption was based on the limited data available from the few expeditions that had reached Hadal depths, and on the extrapolation of the trends observed at shallower depths, where the abundance and diversity of organisms decrease with increasing depth. The extrapolation predicted that the Hadal Zone would contain fewer organisms and fewer species than the abyss, a prediction that has been overturned by the baited camera systems and the lander technology that have revealed vigorous, abundant, and diverse communities at the bottom of the deepest trenches. The overturning of the lifeless hadal assumption is one of the most significant revisions in the history of deep ocean biology because it demonstrates that the deepest zone of the ocean is not a biological wasteland, but a unique biological world with organisms whose biology is strange and specific and precisely adapted to the extreme conditions. The revision has implications for every aspect of deep ocean science, from the estimation of global biodiversity to the prediction of the effects of climate change on the deep ocean ecosystem. The strangeness of the hadal biology is not limited to the molecular adaptations that allow the organisms to function at extreme pressures. It extends to the ecology of the hadal communities, which differs from the ecology of the abyssal communities in ways that reflect the unique food supply dynamics, the unique geographic isolation, and the unique evolutionary history of the hadal zone. The hadal communities are not miniature versions of abyssal communities. They are distinct communities with their own species, their own food webs, and their own ecological dynamics, shaped by the specific conditions of the trenches they inhabit. Now, let us begin. The physical conditions of the hadal zone are defined by three factors that act simultaneously on every organism: extreme pressure, geographic isolation, and a unique food supply. Each factor shapes the biology of the hadal organisms in specific ways, and the combination of all three produces the strange biology that distinguishes the hadal fauna from every other community on the planet. The pressure in the hadal zone ranges from roughly 600 atmospheres at the upper boundary of 6,000 meters to more than 1,100 atmospheres at the bottom of the Challenger Deep, the deepest point in the ocean at approximately 10,935 meters. The pressure increases linearly with depth at a rate of roughly 1 atmosphere per 10 meters, and every organism at every depth in the hadal zone must maintain the function of its molecular machinery against the compressive force that the pressure imposes. The pressure affects every molecular process in the cell, including the folding of proteins, the fluidity of membranes, the rate of enzymatic reactions, and the stability of the nucleic acids that carry the genetic information. The most important molecular adaptation to pressure in the hadal organisms is the accumulation of trimethylamine N oxide, known as TMAO, a small organic molecule that stabilizes proteins against the destabilizing effects of pressure. TMAO works by occupying the space around the protein molecule and by resisting the compression that the pressure would otherwise impose on the protein's structure, maintaining the protein in its functional conformation at pressures that would cause the unmodified protein to denature. The concentration of TMAO in the tissues of deep-sea fish increases linearly with depth, creating a chemical depth gauge that tracks the pressure at each depth and that provides the stabilization needed for the proteins to function at each specific pressure. The TMAO depth gradient is one of the most remarkable findings in hadal biology because it reveals a direct quantitative relationship between the chemistry of the organism and the physics of its environment. The relationship is not approximate. It is linear, with the TMAO concentration increasing by a specific amount for each additional 100 meters of depth, as if the organism's molecular chemistry is precisely calibrated to the pressure at its specific location. The linearity of the relationship has been confirmed by the analysis of fish tissues collected from a range of depths, from the surface to the deepest locations where fish have been found. But here is the problem. The TMAO depth gradient predicts a maximum depth at which fish can survive. The TMAO concentration that is needed to stabilize the proteins at a given depth increases with depth, but the maximum concentration of TMAO that the organism can accumulate is limited by the osmotic pressure that the TMAO generates in the tissues. At a certain depth, the TMAO concentration needed for protein stabilization reaches a level at which the osmotic pressure of the TMAO equals the osmotic pressure of the surrounding seawater, and any further increase in TMAO would cause the cells to lose water to the environment, dehydrating the tissues and killing the organism. The depth at which this osmotic limit is reached has been calculated at roughly 8,200 meters, and this depth corresponds closely to the observed maximum depth at which fish have been found. The prediction is striking. The deepest fish, the Mariana Hadal snailfish, has been recorded at 8,178 meters, just below the predicted maximum. No fish has been recorded below 8,400 meters, despite extensive sampling at greater depths. The absence of fish below the predicted limit is one of the most precise confirmations of a biological prediction in the history of deep ocean science, and it demonstrates that the molecular chemistry of the fish, specifically the osmotic properties of the TMAO, sets a hard limit on the depth at which fish can survive. 3 comparisons establish the scale of the biological strangeness of the Hadal zone. The first concerns the TMAO concentration itself. A surface fish has a TMAO concentration of roughly 40 millimoles per kilogram. A Hadal snailfish at 8,000 meters has a TMAO concentration of roughly 400 millimoles per kilogram, 10 times higher than the surface fish. The tenfold increase in TMAO concentration is the molecular price of living at Hadal depth, a price that is paid by every cell in the body, and that reflects the intensity of the selective pressure for protein stabilization at extreme pressures. The second concerns the creatures that replace fish below the fish depth limit. Below 8,200 meters, the dominant large organisms are the amphipods, small crustaceans that lack the osmotic constraints of fish, and that can accumulate pressure-stabilizing molecules without the osmotic penalty that limits fish. The Hadal amphipods have been collected at depths exceeding 10,000 meters in the deepest parts of the Mariana Trench, where no fish can survive. The amphipods are the apex macrofauna of the deepest ocean, occupying the ecological niche that fish occupy at shallower depths, and their dominance below the fish depth limit is a direct consequence of their different molecular chemistry, which is not subject to the same osmotic limitation. The third concerns the geographic isolation. The Hadal trenches are separated from each other by the abyssal plains, which act as barriers to the dispersal of Hadal organisms. The organisms that inhabit one trench cannot easily reach another trench because the intervening abyssal plain is at a shallower depth and at a lower pressure than the Hadal zone, and the organisms adapted to Hadal pressures cannot survive the lower pressures of the abyss. The isolation has produced endemic species in each trench, species that are found in one trench and nowhere else, species that have been evolving independently since the trench was formed, and species whose biology reflects the specific conditions of their particular trench. The investigation of the specific physical and chemical conditions at different depths within a single trench reveals that the Hadal zone is not uniform, but is structured by gradients of temperature, pressure, chemistry, and food supply that create distinct ecological zones within the trench. The trench walls, which slope from the rim at roughly 6,000 meters to the axis at the maximum depth, provide a depth gradient that supports different communities at different depths. The organisms at the top of the Hadal zone, near 6,000 meters, experience conditions that are intermediate between the abyssal and the deep Hadal, and they include species that are transitional between the two zones. The organisms at the bottom of the trench, at the maximum depth, experience the most extreme conditions and include the most extremely adapted species. The chemical environment within the trench also varies with depth and with distance from the trench walls. The sediment in the trench axis, which accumulates the material that is funneled from the slopes, is rich in organic matter and supports a diverse microbial community. The sediment on the walls, which is steeper and which retains less accumulated material, is poorer in organic matter and supports a less diverse community. The pore water chemistry in the axis sediment differs from the chemistry in the wall sediment, reflecting the different rates of microbial metabolism and the different inputs of organic matter. The investigation of the Hadal microbial community using molecular methods has revealed a diversity and an activity that rival the microbial communities of more productive environments. The bacteria and archaea in the Hadal sediment include species that are unique to the Hadal zone, species whose metabolic capabilities include the degradation of recalcitrant organic compounds, the cycling of nitrogen and sulfur, and the production of methane. The hadal microbial community is not a simplified version of the abyssal microbial community. It is a distinct community with species and metabolic pathways that are adapted to the unique conditions of the trench. The investigation of the nitrogen cycling in the hadal sediment has revealed rates of nitrogen fixation and denitrification that are higher than the rates on the adjacent abyssal plain, reflecting the higher organic matter content of the trench sediment and the more active microbial community. The enhanced nitrogen cycling in the trenches may have implications for the global nitrogen budget, because the trenches, despite their small area, may process nitrogen at rates that are significant when extrapolated to the global total of all hadal trenches. The investigation of the sulfur cycling in the hadal sediment has revealed active sulfate reduction in the deeper layers of the sediment, producing hydrogen sulfide that supports a chemosynthetic microbial community. The chemosynthetic community is similar in function, though not in species composition, to the chemosynthetic communities found at hydrothermal vents, deriving its energy from the oxidation of reduced sulfur compounds rather than from the organic matter that supports the heterotrophic community. The presence of both heterotrophic and chemosynthetic communities in the hadal sediment adds to the metabolic diversity of the hadal zone and provides additional food web pathways that are not found at shallower depths. The investigation of the meobenthic community of the hadal zone, the organisms between roughly 32 micrometers and 1 millimeter in size, has revealed a diverse community of nematodes, copepods, and foraminifera that is adapted to the specific conditions of the trench sediment. The nematodes are the most abundant meobenthic organisms in the hadal zone, as they are at shallower depths, but the species composition of the hadal nematode community is distinct from the composition at shallower depths, with endemic species that are found only in the hadal zone and that possess morphological features adapted to the high-pressure environment. The foraminifera of the hadal zone include the xenophyrophores that have been discussed previously, but they also include smaller species that are abundant in the sediment and that contribute to the cycling of carbon and nutrients. The hadal foraminifera include species with agglutinated tests, external shells made of cemented sediment particles that are morphologically distinct from the agglutinated foraminifera at shallower depths, and that may include endemic species whose biology has not been described. The investigation of the hadal holothurians, the sea cucumbers that inhabit the trench floors, has revealed organisms that are adapted to the food-enriched conditions of the trench axis, and that process the organic matter in the sediment at rates that contribute to the overall carbon cycling of the hadal zone. The hadal holothurians include species that are found only in the hadal zone and that possess morphological adaptations for the high-pressure environment, including reduced skeletal elements and increased body water content. The investigation of how the isolation of the hadal trenches affects the genetic structure of the hadal populations reveals patterns that are consistent with the island biogeography of terrestrial systems. The populations in each trench are genetically distinct from the populations in other trenches, with genetic distances that increase with geographic distance, consistent with the limited dispersal between trenches. The genetic data confirm the pattern of endemism that was inferred from the morphological data and provide quantitative estimates of the degree of isolation and the duration of independent evolution for the populations in each trench. The molecular clock analyses that estimate the divergence times between the populations in different trenches suggest that the isolation has been effective for periods of millions to tens of millions of years, consistent with the geological ages of the trenches and with the tectonic processes that formed them. The divergence times indicate that the hadal populations have been evolving independently for long enough to produce the species-level differences that are observed in the morphological and the genetic data, confirming that the hadal trenches are evolutionary islands that produce new species through the independent evolution of isolated populations. the investigation of the dispersal mechanisms that allow hadal organisms to colonize new trenches, despite the isolation that prevents routine exchange, reveals that the dispersal occurs on geological timescales through rare events that transport organisms across the abyssal barrier. The dispersal mechanisms may include the transport of larvae by deep ocean currents, the colonization of new trenches as they form at subduction zones, and the survival of organisms during brief exposures to the lower pressures of the abyssal plain during transport between trenches. The rare dispersal events create a pattern of relatedness among the hadal fauna in different trenches that reflects the geographic proximity and the geological history of the trenches. The trenches that are closest together tend to share more species and more closely related populations, reflecting the greater probability of dispersal between nearby trenches. The trenches that are most isolated, separated by the greatest distances of abyssal plain, tend to have the most distinctive fauna, reflecting the longer periods of independent evolution. The investigation of whether the hadal zone is expanding or contracting in response to geological and environmental changes reveals that the total area of hadal habitat on the planet is determined by the tectonic processes that create and destroy trenches. New trenches are created as tectonic plates converge and one plate begins to subduct beneath the other. Existing trenches are modified as the subduction process changes the angle of the descending plate, the rate of convergence, and the geometry of the trench. The total area of hadal habitat is therefore not fixed, but changes over geological timescales, with new trenches appearing and old trenches being modified or eliminated as the tectonic plates move. The investigation of how the environmental changes driven by climate change are affecting the hadal zone reveals that the hadal communities are not immune to the effects of surface changes, despite the great depth that separates them from the surface. The warming of the surface ocean is altering the stratification of the water column and may be reducing the marine snow flux that feeds the hadal community. The deoxygenation of the intermediate waters is reducing the oxygen available to the hadal organisms, and the acidification of the deep water is changing the chemistry of the hadal environment in ways that may affect the calcification of the foraminifera and the chemistry of the pore water. The detection of microplastics in the hadal zone, documented by researchers who analyze sediment samples from the Mariana Trench and other hadal locations, demonstrates that the human impact on the deep ocean extends to the most remote habitats on the planet. The microplastics, which are fragments of plastic debris that have sunk from the surface, have been found in the gut contents of hadal amphipods, indicating that the organisms are consuming the plastic and that the plastic is entering the hadal food web. The health effects of microplastic consumption on the hadal organisms are unknown, but are a concern because the plastics may carry toxic chemicals and because the mechanical effects of the plastic fragments on the gut tissue may impair the organism's ability to process food. The presence of microplastics at hadal depths is one of the most dramatic illustrations of the global reach of human pollution and of the vulnerability of even the most remote habitats to the effects of human activity. The hadal zone, which was once considered too remote to be affected by surface activities, is now known to contain human-made pollutants, including plastics, persistent organic pollutants, and heavy metals that have been transported to the deepest ocean by the same physical processes that deliver the marine snow and the food debris that sustain the hadal community. The protection of the hadal zone from human impact requires the recognition that the hadal communities are unique, that they contain endemic species that are found nowhere else, that they possess biology that is found in no other habitat, and that they are vulnerable to disturbance from which they may not recover. The hadal communities have been evolving in isolation for millions of years, producing organisms that are adapted to the most extreme conditions on the surface of the Earth. The disruption of these communities through pollution, mining, or the cascading effects of climate change would be the disruption of biological experiments that have been running for geological timescales and that have produced results, the strange and precisely adapted organisms of the hadal zone that cannot be replicated by any human process. The Hadal Zone is the most extreme biological habitat on the planet. Its organisms possess the strangest biology. Its communities contain the highest endemism. Its food supply is paradoxically enriched by the funnel geometry of the trenches. Its isolation is enforced by the pressure barrier of the abyssal plain. And its biology, when examined at the molecular level, reveals adaptations that are as precise, as comprehensive, and as extraordinary as any that the evolutionary process has produced on the surface of the Earth. The strange biology of Hadal Zone creatures is the deep ocean's final lesson in the capacity of life to adapt to extreme conditions. The lesson is that life finds a way, even at the most extreme pressures, even in the most complete darkness, even in the most isolated habitats, even at the molecular limits of what biological chemistry can achieve. The lesson is taught by the organisms themselves, by their TMAO-saturated proteins, by their unsaturated membranes, by their pressure-optimized enzymes, by their rewritten genomes, by their symbiotic gut bacteria, by their earthquake-delivered food, and by their strange, beautiful, precisely adapted bodies that thrive at the bottom of the deepest gashes in the surface of the Earth. The Hadal Zone is not lifeless. It has never been lifeless. It is one of the most biologically active and most biologically distinctive habitats on the planet, and its organisms, strange and precisely adapted, are the most extreme products of the evolutionary process, living their extraordinary lives at the molecular limits of life, in the deepest darkness, under the greatest pressure, in the most isolated habitats on the surface of the world. The food supply of the Hadal Zone is paradoxically richer than the food supply of the adjacent abyssal plains, despite the greater depth. The V-shaped geometry of the trenches acts as a funnel, concentrating the organic matter that sinks from the surface into the narrow trench axis. The marine snow that would be distributed across a wide area of the abyssal plain is funneled into the trench, producing a food supply per unit area that can be several times higher than the food supply on the plain. The enrichment is amplified by the earthquakes that periodically shake the trench walls, dislodging accumulated sediment and organic matter from the slopes and delivering it to the trench axis in turbidity currents, rapid flows of sediment-laden water that carry concentrated organic material to the deepest parts of the trench. The Hadal food funnel creates conditions that support biological communities that are more abundant and more active than the communities on the adjacent abyssal plains, a finding that was surprising when it was first documented by baited camera systems deployed at Hadal depths. The baited cameras attracted swarms of amphipods within hours of deployment, with hundreds of individuals arriving at the bait within the first day, a response that indicated a resident population that was both large and metabolically active, far more active than the sparse, slow-moving communities of the abyssal plain. The Hadal communities are not the impoverished remnants of abyssal communities that have been pushed to the extreme. They are vigorous, active, and well-fed communities that are sustained by the food funnel, and that include organisms whose biology is specifically adapted to the unique conditions of the trench. The core problem with the strange biology of Hadal Zone creatures is that the conditions in the Hadal impose constraints on every molecular process in the cell, and the organisms must simultaneously solve multiple molecular problems that are each at the extreme edge of what biological chemistry can accommodate. The protein folding problem is the most fundamental. Proteins are molecular machines that must fold into specific three-dimensional shapes to perform their functions. The folding is driven by the interactions between the amino acids that compose the protein, and the correct folding depends on the balance of forces, including hydrogen bonds, van der Waals forces, and the hydrophobic effect that stabilize the native structure. Pressure disrupts this balance by compressing the protein, reducing the volume of the internal cavities that are essential for the protein's structure, and forcing the protein into a more compact conformation that may not be functional. The TMAO counteracts this disruption by increasing the energetic cost of the unfolded state, making the folded state thermodynamically more favorable and maintaining the protein in its functional conformation. And it gets worse. The membrane fluidity problem is equally fundamental. The cell membrane, a bilayer of lipid molecules that encloses the cell and that controls the transport of molecules into and out of the cell, must maintain a specific fluidity to function correctly. Too fluid, and the membrane loses its structural integrity. Too rigid, and the membrane proteins cannot move within the bilayer, impairing their function. Pressure increases the rigidity of the membrane by compressing the lipid molecules and reducing their freedom of movement. The hadal organisms counteract this rigidity by incorporating unsaturated fatty acids into their membranes, fatty acids whose kinked molecular structures resist compression and maintain fluidity at the pressures of the hadal zone. The specific lipid compositions of hadal organisms have been analyzed from specimens collected at hadal depths, and the analyses reveal membrane compositions that are distinct from those of surface organisms and from those of abyssal organisms. The hadal organisms have higher proportions of unsaturated fatty acids, lower proportions of cholesterol, and unique lipid species that have not been found in organisms from shallower depths. The unique lipid chemistry of the hadal organisms is a molecular adaptation that is as specific to the hadal zone as the TMAO concentration is, and it reflects the intensity of the selective pressure for membrane function at extreme pressures. The enzyme kinetics problem adds another layer of complexity. Enzymes, the protein catalysts that drive every metabolic reaction in the cell, must function at rates that are sufficient for the metabolic demands of the organism. Pressure affects the rate of enzymatic reactions by altering the volume changes that occur during the catalytic cycle, either accelerating or decelerating the reaction, depending on the specific enzyme and the specific step in the catalytic mechanism. The hadal organisms possess enzymes whose kinetic properties are adapted to the pressures of the hadal zone with catalytic rates that are optimized for the specific pressure at the organism's depth, and that differ from the rates of the same enzymes in surface or abyssal organisms. This is not science fiction. These are measured, documented molecular properties of organisms that have been collected from the hadal zone and analyzed in laboratories around the world. The TMAO concentrations, the lipid compositions, and the enzyme kinetics are all published in the peer-reviewed literature, and they collectively paint a picture of organisms whose molecular machinery has been comprehensively remodeled by natural selection for the specific conditions of the hadal zone. But here is what makes this truly terrifying. The genomic analysis of the hadal snailfish has revealed that the organism's genome has been substantially modified compared to the genomes of its shallow water relatives, with specific genes lost, specific genes duplicated, and specific genes under strong positive selection. The lost genes include genes for vision-related proteins that are not needed in the total darkness of the hadal zone, and genes for swim bladder formation that are not needed in an organism that lacks a swim bladder. The duplicated genes include genes for TMAO synthesis and transport, reflecting the increased demand for the pisolite at hadal depths, and genes for DNA repair, reflecting the increased need for genome maintenance under conditions where the molecular damage rate may be elevated. The positive selection on specific genes, identified by the comparison of the hadal snailfish genome with the genomes of related species at shallower depths, reveals the specific molecular targets on which natural selection has acted to produce the hadal adaptations. The targets include the genes for cytoskeletal proteins, which maintain the structural integrity of the cell under pressure, the genes for transport proteins, which maintain the ionic balance of the cell against the osmotic effects of the high TMAO concentration, and the genes for lipid metabolism, which produce the unique fatty acids that are needed for membrane function at hadal pressures. Something is wrong with our assumptions about the uniformity of deep-sea biology. The hadal zone is not merely a deeper version of the abyss. It is a biologically distinct zone, with organisms whose molecular, genomic, and metabolic biology is specifically adapted to the conditions of the trench, and that differs from the biology of abyssal organisms in ways that are as significant as the differences between the biology of terrestrial and aquatic organisms. The hadal zone is a separate biological world, as distinct from the abyss as the abyss is from the surface. The attempts to understand the strange biology of hadal zone creatures have been transformed by the development of land technology, autonomous instruments that can be deployed to the bottom of the deepest trenches and that can collect specimens record video, and make measurements at the pressures of the Hadal zone without the expense and the risk of crude submersible dives. The Hadal landers, developed by research groups at the University of Aberdeen, the University of Western Australia, and the Japan Agency for Marine Earth Science and Technology, among others, are free-falling instruments that sink to the trench floor under their own weight and that are recovered by releasing a ballast weight and floating to the surface. The landers can be equipped with baited cameras that record video of the organisms that are attracted to the bait, with traps that collect live specimens at Hadal pressure, and with sensors that measure the temperature, the pressure, the salinity, and the oxygen concentration of the water. The numbers are striking when the baited camera data from the Hadal landers are analysed. The cameras deployed in the Mariana Trench, the Kermadec Trench, the Tonga Trench, the Japan Trench, and the Peru Chile Trench have documented swarms of amphipods arriving at the bait within hours, with hundreds of individuals visible in a single frame, demonstrating that the Hadal community is far more abundant and far more active than the sparse, slow-moving communities of the abyssal plain. The abundance at the bait is a measure of the local population density and the mobility of the organisms, and both measures indicate that the Hadal communities are vigorous, well-fed, and capable of rapid response to food inputs. The investigation of the Hadal amphipod Hydrothermal gigas, one of the most abundant organisms in the Hadal zone of the Pacific Trenches, has revealed a biology that is strange even by the standards of the Hadal. The Hydrothermal possesses cellulase enzymes that break down cellulose, the structural polymer of plant cell walls. The possession of cellulase enzymes indicates that the Hydrothermal can digest wood, a capability that is rare among marine animals and that has no obvious function in the deep ocean where there are no forests and no plant material. The explanation is that the Hydrothermal's cellulase capability allows it to exploit the wood debris that sinks to the Hadal zone from the surface, the remains of terrestrial plants that are carried to the ocean by rivers and that eventually sink to the deep. The wood debris is a food source that is not exploited by most deep ocean organisms because most lack the enzymes needed to break down the cellulose. The Hydrothermal's cellulase enzymes give it access to a food source that its competitors cannot use, a competitive advantage that may contribute to the organism's abundance in the Hadal zone. The enzymes are believed to be derived from symbiotic bacteria in the amphipod's gut, bacteria that produce the cellulase and that provide the amphipod with the ability to digest a substrate that its own genome cannot process. The gut microbiome of the Hadal amphipods has been investigated using molecular methods and has been found to contain bacterial communities that are distinct from the gut microbiomes of amphipods at shallower depths. The Hadal gut microbiome includes bacteria that are adapted to high pressure, bacteria that produce enzymes for the digestion of recalcitrant organic matter, and bacteria whose metabolic capabilities may include the synthesis of vitamins and amino acids that are not available from the amphipod's diet. The gut microbiome is therefore not merely a passive resident of the amphipod's digestive tract, but an active partner in the organism's nutrition, providing metabolic capabilities that the amphipod's own genome does not encode. The investigation of the piezophilic bacteria of the Hadal zone, bacteria that require high pressure to grow and that cannot grow at surface pressure, has revealed organisms whose biology is as strange as the biology of the macrofauna. The piezophilic bacteria have been isolated from Hadal sediment and from the gut contents of Hadal organisms, and they have been grown in the laboratory in pressure vessels that maintain the high pressures of the Hadal zone. The bacteria grow optimally at pressures of 600 to 1000 atmospheres and do not grow at pressures below roughly 400 atmospheres, indicating that their molecular machinery is not merely adapted to high pressure, but requires high pressure for normal function. The piezophilic bacteria possess membranes with lipid compositions that are radically different from the compositions of surface bacteria, with extremely high proportions of unsaturated fatty acids that maintain the fluidity needed for membrane function at Hadal pressures. The bacteria also possess unique enzymes whose catalytic activities are optimized for high pressure. with reaction rates that increase with pressure rather than decreasing, the opposite of the pattern seen in surface enzymes. The piezophilic bacteria are the most extreme pressure-adapted organisms on the planet, and their biology provides insights into the fundamental mechanisms by which living systems can be adapted to extreme physical conditions. The math is striking when the biomass of the hadal communities is compared with the biomass of the adjacent abyssal communities. The hadal trench communities, despite occupying a much smaller area, can have biomass densities that are several times higher than the densities on the adjacent abyssal plain, reflecting the food funnel effect that concentrates organic matter in the trench axis. The higher biomass density in the trenches challenges the assumption that the deepest ocean is the most impoverished, and it demonstrates that the geometry of the habitat, the V-shaped trench that funnels food from the slopes to the axis, can create conditions that are more favorable for life than the conditions at shallower depths on the flat abyssal plain. The investigation of the xenophyophores, giant single-celled organisms that inhabit the walls and the floor of the hadal trenches, has revealed some of the strangest organisms in the hadal zone. The xenophyophores can reach diameters of more than 10 centimeters, making them the largest single-celled organisms in the ocean. The organisms construct tests, external shells made of agglutinated particles of sediment and organic detritus that protect the cell and that provide a surface for the attachment of smaller organisms. The xenophyophores are abundant on the walls of the hadal trenches, forming dense aggregations that modify the local sediment surface and that create microhabitats for other organisms. The biology of the xenophyophores is poorly understood because the organisms are difficult to collect intact and because they do not survive the decompression that occurs during recovery from hadal depths. The organisms are believed to feed on the organic matter in the sediment, extracting nutrients from the particles that they incorporate into their tests, and they may also harbour symbiotic bacteria within their cytoplasm that contribute to their nutrition. The xenophyophores of the hadal zone are larger and more abundant than the xenophyophores at shallower depths, suggesting that the food funnel effect provides a food supply that supports larger cell sizes and higher population densities than the abyssal plain. The investigation of the role of earthquakes in the hadal food supply has revealed that seismic events can deliver large pulses of organic matter to the trench axis, creating episodic food bonanzas that supplement the continuous rain of marine snow. The earthquakes that shake the trench walls dislodge accumulated sediment and organic matter, sending turbidity currents, flows of sediment-laden water, racing down the slopes and into the axis. The turbidity currents deliver concentrated organic material to the deepest parts of the trench, creating a food pulse that is consumed by the resident community over a period of weeks to months. The frequency and the magnitude of the earthquake-driven food pulses vary among trenches, reflecting the seismic activity of each trench, and the variation may contribute to the differences in community structure and abundance that have been observed among different trenches. The deeper truth about the strange biology of hadal zone creatures is that the hadal zone is not an extension of the deep ocean. It is a different biological world, separated from the abyss by a boundary that is defined by the molecular chemistry of the organisms that inhabit it. The boundary is not a line on a map. It is a molecular transition, a depth at which the concentration of pressure-stabilizing molecules reaches a level that requires specific genomic, metabolic, and physiological adaptations that are not found in abyssal organisms. The transition occurs at roughly 6,000 meters, the depth at which the TMAO concentration, the lipid composition, and the enzyme kinetics of the organisms begin to diverge significantly from the values found in abyssal organisms. Let that sink in. The hadal zone is not merely deep. It is biologically distinct, with organisms whose molecular machinery has been remodeled by natural selection for the specific conditions of the trench. The remodeling is comprehensive, affecting every category of biological molecule, every metabolic pathway, and every physiological system. The organisms are not surviving at hadal depth. They are adapted to hadal depth as thoroughly as any organism is adapted to any environment on the planet. Let that sink in. This is not science fiction. This is the documented biology of the Hadal Zone, based on the molecular analysis of specimens collected from the deepest trenches in the ocean and on the genomic analysis of the Hadal organism's DNA. The adaptations are real. The molecular changes are measured. The genomic modifications are sequenced. And the biological distinctiveness of the Hadal Zone is confirmed by every line of evidence that has been examined. The deeper truth extends to the evolutionary implications of the Hadal Zone's geographic isolation. Each trench is an island of extreme conditions, separated from other trenches by the barrier of the abyssal plain. The organisms in each trench have been evolving independently since the trench was formed, producing endemic species that are found in one trench and nowhere else. The endemism is high, with studies suggesting that 30 to 50% or more of the species in each trench are endemic, found only in that trench. The endemism reflects the millions of years of independent evolution that the isolation has permitted, and it demonstrates that the Hadal trenches are evolutionary laboratories, producing new species through the same processes of mutation, selection, and genetic drift that produce new species in every isolated habitat. The comparison of the Hadal communities in different trenches reveals patterns of similarity and difference that reflect the geographic proximity, the geological age, and the environmental conditions of each trench. The trenches that are geographically close tend to share more species than the trenches that are far apart, reflecting the greater probability of dispersal between nearby trenches. The trenches that are older tend to have more endemic species than the younger trenches, reflecting the longer period of independent evolution. And the trenches that have different environmental conditions, including different temperatures, different food supplies, and different sediment compositions, tend to have communities that are adapted to the specific conditions of each trench. Something is wrong with our assumptions about the uniformity of the deep ocean fauna. The Hadal Zone demonstrates that the deep ocean is not a single, uniform habitat, but a collection of distinct environments, each with its own conditions, its own community, and its own evolutionary history. The assumption that the deep ocean fauna is uniform across all depths and all locations is incorrect, and the Hadal Zone is the most dramatic demonstration of this incorrectness, with a fauna that is as distinct from the abyssal fauna as the fauna of one continent is from the fauna of another. The implications for astrobiology connect the Hadal biology to the search for life on other worlds. The Hadal organisms demonstrate that life can function at pressures exceeding 1,000 atmospheres, can adapt its molecular machinery to extreme physical conditions, and can thrive in isolated environments that are separated from the rest of the biosphere by barriers that are effectively impassable. These capabilities are relevant to the search for life in the high-pressure environments that are expected to exist in the interiors of icy moons and in the deep subsurface of rocky planets, environments where the conditions may be analogous to the conditions in the Hadal Zone, and where the adaptations that the Hadal organisms possess may be the adaptations that extraterrestrial life would need. What does it mean that the deepest trenches in the ocean contain organisms whose biology is fundamentally different from the biology of the rest of the deep ocean? What does this tell us about the capacity of living systems to adapt to extreme conditions, about the relationship between geography and evolution, and about the limits of what biological molecules can achieve? The first implication is about the precision of biological adaptation. The linear relationship between TMAO concentration and depth, the specific lipid compositions at each pressure, and the pressure-optimized enzyme kinetics of the Hadal organisms all demonstrate that the molecular machinery of life can be precisely calibrated to the physical conditions of the environment, with adaptations that track the environmental parameters with quantitative accuracy. The calibration is not approximate, it is precise, with the TMAO concentration increasing by a specific amount for each additional 100 meters of depth, as if the organism has a molecular pressure gauge that sets the chemistry to the correct level for each specific depth. The second implication concerns the hard limits of biological adaptation. The TMAO osmotic limit that prevents fish from surviving below roughly 8,200 meters is a hard limit set by the fundamental properties of the TMAO molecule and of the cell membrane that cannot be exceeded by any adaptation within the fish body plan. The limit demonstrates that the capacity of living systems to adapt to extreme conditions has boundaries that are set by the fundamental properties of the molecules that compose the living system and that cannot be exceeded regardless of the evolutionary time available. There is a philosophical pause worth taking here. The Hadal trenches have been producing their strange organisms for tens of millions of years, in isolation, under pressures that compress every molecule, in darkness that has no beginning and no end. The organisms that inhabit the trenches have been shaped by these conditions into forms and biochemistries that have no equivalent anywhere else on the planet. The trenches are the most extreme biological laboratories on Earth, and the organisms they produce are the most extreme products of the evolutionary process, pushed to the molecular limits of what life can achieve. The creatures of the Hadal zone deserve specific reflection. The Mariana snailfish, with its gelatinous body and its TMAO-saturated tissues, surviving at 8,178 meters, just below the predicted maximum depth for fish. The supergiant amphipod Alicella gigantea, reaching lengths exceeding 34 centimeters, thriving in the deepest trenches where no fish can follow. The Hirondelia gigas, with its cellulase enzymes and its symbiotic gut bacteria, digesting wood at the bottom of the Pacific trenches. The piezophilic bacteria, requiring pressures of 600 atmospheres to grow, with metabolisms that cannot function at the surface. The xenophytophores, single cells 10 centimeters across, building their agglutinated tests from the particles of the trench floor. Each is a product of the Hadal zone, shaped by its specific conditions, and each possesses biology that is found nowhere else on the planet. The conservation of the Hadal zone communities is a concern that is becoming increasingly urgent as human activities extend into the deepest parts of the ocean. The potential for deep-sea mining in the Hadal zone, while currently limited by the technical difficulty of operating at extreme depths, is a future possibility that would threaten the endemic communities of the trenches. The disposal of waste in the Hadal zone, including the disposal of munitions and industrial waste that has occurred in several trenches, has already introduced contaminants to the Hadal environment with unknown consequences for the organisms that inhabit it. The strange biology of Hadal zone creatures is the biology of life at its limits. The organisms that inhabit the Hadal zone are adapted to the most extreme pressures on the surface of the Earth through molecular modifications that are as precise as they are comprehensive. The TMAO that stabilizes their proteins, the unsaturated lipids that maintain their membranes, the pressure-optimized enzymes that drive their metabolism, the rewritten genomes that encode the molecular machinery needed for life at the extreme. Each adaptation is a specific solution to a specific physical constraint, and together they constitute a comprehensive biological engineering package that allows life to function at pressures that would disable every surface organism. The Hadal zone is the last frontier of biology on this planet. Its organisms are the most extreme products of the evolutionary process. Its conditions are the most demanding on the surface of the Earth, and its biology, strange and specific and precisely adapted, is a testament to the capacity of living systems to find solutions to physical problems that seem, by every surface standard, unsolvable. The fish depth limit at roughly 8,200 meters is the most dramatic expression of the Hadal zone's strangeness, a hard boundary set by the molecular chemistry of the TMAO and enforced by the fundamental properties of the cell membrane. Below this depth, fish cannot survive, and the ecological niche that fish occupy at shallower depths is filled by the amphipods, the crustaceans whose different molecular chemistry allows them to function at pressures that exceed the fish limit. The transition from fish to amphipod dominance at 8,200 meters is one of the sharpest ecological boundaries in the ocean, a boundary that is set not by competition or by predation, but by the molecular properties of the organisms themselves. The Hadal zone is not merely deep, it is biologically distinct, it is geographically isolated, It is food enriched by the funnel geometry of the trenches. It is seismically disturbed by the earthquakes that shake the trench walls. And it is populated by organisms whose biology is as strange, as specific, and as precisely adapted as any biology on the planet. The strange biology of the Hadal Zone is the deep ocean's most extreme demonstration of what life can achieve at the molecular limits of adaptation. The organisms are there, in the trenches, at the bottom of the deepest gashes in the surface of the Earth, living their strange lives with their strange molecules in their strange habitats, adapted to conditions that no other organism on the planet can tolerate, thriving in an environment that was once thought to be lifeless. The Hadal Zone is not lifeless. It has never been lifeless. It is full of life, strange life, precisely adapted life, life that is pushed to the molecular limits of what biological chemistry can achieve. The organisms are there. They have been there for millions of years. And their biology, when examined in detail, reveals the most extreme and the most precise adaptations that the evolutionary process has ever produced on the surface of the Earth. The deepest trenches contain the strangest biology. The strangest biology reveals the limits of life, and the limits, as the Hadal organisms demonstrate, are further than anyone imagined before the landers descended, and the specimens came up, and the molecules were measured. The Hadal Zone is the edge of life. The organisms that inhabit it are the explorers of that edge, and the biology they possess, strange and precise and adapted beyond any surface expectation, is the most compelling evidence that life, pushed to its limits, finds ways to persist that the standard models of biology did not predict, and that the study of the surface world could never have revealed. The specific molecular mechanisms by which TMAO stabilizes proteins under pressure deserve expanded treatment, because they reveal the precision of the molecular adaptation, and because they illustrate a general principle of how small molecules can modulate the physical properties of biological macromolecules. TMAO is an osmolyte, a small organic molecule that affects the properties of water and, through the water, the properties of the proteins that are dissolved in it. TMAO increases the surface tension of the water around the protein, making the exposure of the protein's hydrophobic interior to the water more energetically costly. The increased cost of exposure stabilizes the folded state of the protein, in which the hydrophobic interior is buried, relative to the unfolded state, in which the interior is exposed. The stabilization is proportional to the concentration of the TMAO, which is why the TMAO concentration increases linearly with depth, tracking the increasing pressure that would otherwise destabilize the proteins. The TMAO mechanism is not unique to the Hadal organisms. TMAO is found in the tissues of marine organisms at all depths, but its concentration increases with depth in a pattern that has been documented across multiple families of deep-sea fish. The relationship between TMAO concentration and depth was first described by the biochemist Paul Yancey at Whitman College, who analyzed the tissues of deep-sea fish collected from a range of depths, and who demonstrated the linear relationship that has become one of the foundational findings of Hadal biology. Yancey's work established the TMAO depth gauge as a general principle of deep ocean biochemistry and predicted the fish depth limit at roughly 8,200 meters, a prediction that has been confirmed by the subsequent collection of fish from near maximum depths and by the absence of fish from greater depths. The investigation of the Hadal snailfish genome by a research team led by scientists at the Chinese Academy of Sciences produced one of the most detailed molecular portraits of a Hadal organism. The genome was sequenced from specimens collected in the Mariana Trench at depths exceeding 7,000 meters, and the analysis revealed a genome that has been substantially modified compared to the genomes of related snailfish species at shallower depths. The modifications include the loss of several gene families that are no longer functional in the Hadal environment. The genes for opsins, the visual pigments that detect light, have been reduced in number, consistent with the total darkness of the Hadal zone. The genes for osteocalcin, a protein involved in bone mineralization, have been modified, consistent with the reduced skeletal mineralization that characterizes the Hadal snailfish, whose bones are poorly calcified and whose body is correspondingly soft and flexible. The reduced skeletal mineralization is an adaptation to the high pressure because the reduced mineral content reduces the density of the body, and because the flexibility of the poorly mineralized skeleton allows the body to compress without fracturing under the ambient pressure. The modifications also include the amplification of gene families that are important for hadal life. The genes for TMAO biosynthesis and transport have been duplicated, providing the genomic capacity for the high TMAO production that is needed at hadal pressures. The genes for fatty acid desaturation, the enzymes that convert saturated fatty acids to unsaturated fatty acids, have been amplified, providing the capacity for the production of the unsaturated lipids that are needed for membrane fluidity at hadal pressures. And the genes for DNA repair enzymes have been amplified, providing enhanced capacity for the maintenance of the genome against the molecular damage that accumulates under the extreme conditions. The positive selection analysis of the hadal snailfish genome identified specific amino acid changes in specific proteins that are believed to be adaptive for the hadal environment. The changes include substitutions in the cytoskeletal proteins that maintain the structural integrity of the cell, substitutions that may increase the stability of the cytoskeleton under pressure. The changes also include substitutions in the ion channel proteins that control the flow of ions across the cell membrane, substitutions that may compensate for the effects of pressure on the ion channel's gating mechanism, and that may maintain the normal electrical signaling of the nerve and muscle cells at hadal pressures. The investigation of the hadal amphipod's biology has revealed adaptations that are distinct from the adaptations of the hadal fish, and that reflect the different body plan and the different molecular chemistry of the crustacean lineage. The amphipods do not use TMAO as their primary pressure stabilizing molecule. Instead, they use a combination of free amino acids and other organic osmolytes that provide the protein stabilization needed for function at hadal pressures. The different molecular strategy is the reason that amphipods are not subject to the same osmotic limitation that prevents fish from surviving below 8,200 meters, because the osmolytes used by the amphipods do not generate the same osmotic pressure as the TMAO and can be accumulated to higher concentrations without dehydrating the cells. The supergiant amphipod Alicella gigantea, which can reach lengths exceeding 34 centimeters, is one of the largest amphipods in the ocean and one of the most striking examples of deep-sea gigantism. The organism is found at hadal depths in several trenches and is a scavenger that feeds on the carcasses of fish and other organisms that sink to the trench floor. The organism's large body size is believed to be an adaptation to the food pulse dynamics of the hadal zone because a large body can store more energy than a small body and can therefore survive longer between food inputs. The large body size also allows the organism to travel greater distances to reach food falls, increasing the probability of encountering food in the vast volume of the trench. The investigation of the hadal amphipod Hirondelia gigas has revealed additional metabolic capabilities that reflect the unique food supply of the hadal zone. In addition to the cellulase enzymes that allow it to digest wood, the Hirondelia possesses enzymes for the degradation of other recalcitrant organic compounds, including lignin and chitin, that are resistant to decomposition by most marine organisms. The ability to degrade these compounds gives the Hirondelia access to food sources that other organisms cannot use and may explain its dominance in the hadal amphipod community, where it is often the most abundant macrofaunal organism. The gut microbiome of Hirondelia gigas has been characterized using molecular methods and has been found to contain a diverse community of bacteria that includes cellulose-degrading species, lignin-degrading species, and species that produce essential vitamins and amino acids. The microbiome is structured by the pressure and the chemistry of the hadal environment, with bacterial species that are adapted to the conditions of the trench, and that differ from the gut bacteria of amphipods at shallower depths. The microbiome is believed to be acquired from the environment rather than transmitted from parent to offspring, which means that each individual must assemble its gut community from the bacteria available in its specific trench. potentially contributing to the trench-specific differences in amphipod biology that have been observed. The investigation of the Hadal zone's role in the global carbon cycle has revealed that the trenches may sequester carbon at rates that are higher than the rates on the adjacent abyssal plains. The food funnel effect that concentrates organic matter in the trench axis increases the rate at which carbon is delivered to the trench, and the burial of this carbon in the trench sediment, which is rapidly accumulating due to the input of material from the slopes and from the earthquake-driven turbidity currents, may sequester the carbon for geological timescales. The trenches may therefore be more important for carbon sequestration than their small area would suggest, a possibility that has implications for the estimation of the global ocean's carbon budget. The investigation of the specific trench-by-trench differences in the Hadal fauna provides a framework for understanding how the geographic isolation of the trenches has produced distinct biological communities. The Mariana Trench, the deepest trench in the ocean, contains the Hadal snailfish Pseudoliparis swiri, a species that has not been found in any other trench. The Kermadec Trench, south of New Zealand, contains a different species of Hadal snailfish that is morphologically and genetically distinct from the Mariana species. The Japan Trench contains a community of Hadal organisms that is adapted to the specific conditions of the northwestern Pacific, including the high seismic activity that delivers frequent food pulses to the trench floor. The trench-by-trench comparison reveals that the Hadal fauna is not a single community, but a collection of distinct communities, each adapted to the specific conditions of its trench, and each containing endemic species that are found nowhere else. The endemism is highest in the most isolated trenches and in the deepest zones of each trench, where the barrier to dispersal is the greatest. The pattern of endemism is consistent with the predictions of island biogeography theory, which predicts that isolated habitats should contain more endemic species than connected habitats, and it confirms that the Hadal trenches function as biological islands in a sea of abyssal plain. The 5 Deeps expedition of 2018 to 2019, led by the explorer Victor Vescovo, visited the deepest point of each of the 5 oceans, using the submersible limiting factor, providing the first comprehensive visual survey of the deepest habitats on the planet. The expedition documented organisms at every location, including amphipods, holothurians, and unidentified organisms, confirming that the Hadal zone contains life at every depth and in every ocean basin. The expedition also collected water and sediment samples for molecular analysis, providing environmental DNA data that are being used to characterize the microbial and macrofaunal communities of the deepest trenches. The Trieste descent of 1960, in which the Swiss engineer Jacques Piccard and the American naval officer Don Walsh descended to the bottom of the Challenger Deep in the Bathyscaphe Trieste, was the first human visit to the Hadal zone and provided the first direct observation of life at Hadal depth. Piccard reported seeing a flat fish-like organism on the bottom of the trench, an observation that was debated for decades because some biologists doubted that a fish could survive at that depth. The debate was resolved by the subsequent discovery of the Hadal snailfish and by the deployment of baited cameras that documented fish at depths exceeding 7,000 meters, confirming that fish are present in the Hadal zone, though the specific organism that Piccard observed has never been definitively identified. The investigation of the Hadal zone's potential as a source of novel biotechnological products has identified several categories of molecules that are of interest to the pharmaceutical and industrial biotechnology sectors. The pressure-stable enzymes of the piezophilic bacteria, which function at pressures that would inactivate conventional enzymes, are of interest for industrial processes that are conducted at high pressure, including the synthesis of polymers and the processing of food. The unique lipids of the Hadal organisms, which maintain their fluidity at extreme pressures, are of interest as components of liposomal drug delivery systems that must maintain their integrity under the pressures encountered in the human body. The TMAO and other osmolytes of the Hadal organisms are of interest as protein-stabilizing agents for the pharmaceutical industry, where the stability of protein-based drugs during manufacturing and storage is a significant concern. The Hadal zone is the most extreme biological habitat on the surface of the Earth. Its organisms are the most extremely adapted. Its conditions are the most demanding. Its isolation is the most complete. And its biology, when examined in molecular detail, reveals adaptations that are as precise, as comprehensive, and as strange as any that the evolutionary process has produced on this planet. The organisms of the Hadal zone are not merely surviving at extreme depth. They are adapted to it, thriving in it, and demonstrating through their strange and precise biology that the limits of life are set not by the depth of the water, but by the molecular properties of the organisms themselves. The limits are further than anyone imagined before the landers descended, before the specimens were analysed, and before the molecules were measured, and the Hadal zone, dark and deep and pressurised, continues to reveal new adaptations and new organisms with every expedition that descends to its depths. The Hadal zone is the edge of life on the surface of the Earth. The organisms that inhabit it are the explorers of that edge, and the strange biology that they possess is the most compelling evidence that life, pushed to its molecular limits, finds solutions that are as extraordinary as the conditions that demand them. The investigation of the specific pressure effects on biological processes at the molecular level provides the theoretical foundation for understanding why the Hadal biology is so strange. Pressure affects biological molecules through the principle of Le Chatelier, which states that a system under pressure will shift toward the state that occupies the smallest volume. For protein folding, the unfolded state typically occupies a smaller volume than the folded state because the unfolding allows water molecules to fill the cavities and voids within the protein's interior. Pressure, therefore, favors the unfolded state, destabilizing the protein and pushing it toward denaturation. The TMO counteracts this effect by making the unfolded state energetically more costly, shifting the equilibrium back toward the folded state and maintaining the protein's function. The effect of pressure on enzyme catalysis is more complex, because the catalytic cycle involves multiple steps, each with its own volume change. The binding of the substrate to the enzyme, the formation of the transition state, the release of the product, and the resetting of the enzyme all involve changes in the volume of the enzyme-substrate complex. And pressure affects each step according to its specific volume change. The net effect of pressure on the overall rate of catalysis depends on which step has the largest volume change and whether that change is positive or negative. For some enzymes, the net effect is inhibitory, with pressure slowing the reaction. For others, the net effect is activating, with pressure accelerating the reaction. The Hadal enzymes have been selected for catalytic cycles in which the net effect of pressure is either neutral or activating, maintaining the enzymatic rates that are needed for the metabolic demands of the organism at Hadal pressures. The investigation of the Hadal organism's DNA repair systems has revealed an enhanced capacity for the correction of DNA damage that is consistent with the prediction that the extreme conditions of the Hadal zone may increase the rate of molecular damage. The DNA damage in the Hadal organisms may be increased by the direct effects of pressure on the DNA structure, which can cause the unwinding and the denaturation of the double helix, and by the indirect effects of the reactive oxygen species that may be produced at higher rates under pressure. The enhanced DNA repair capacity, documented through the amplification of repair-related genes in the Hadal snailfish genome, provides the organisms with the molecular tools needed to maintain their genomic integrity against the increased damage rate. The investigation of the reproductive biology of the Hadal organisms has revealed strategies that reflect the extreme conditions and the isolation of the Hadal zone. The Hadal snailfish are believed to be semelparous, reproducing once and then dying, a strategy that is consistent with the short lifespan and the rapid growth rate that have been inferred from the statolith ring counts of deep-sea snailfish. The semelparous strategy concentrates all of the organism's reproductive investment into a single event, maximizing the number of offspring produced and the probability that some will survive to reproduce in the next generation. The Hadal amphipods are believed to have a different reproductive strategy, with multiple reproductive events during their lifespan, a strategy that is consistent with the longer lifespan and the slower growth rate that have been inferred for these organisms. The female amphipods brood their eggs in a marsupium, a pouch on the underside of the body, and the developing embryos are protected from the pressure and the predation of the hadal environment until they are large enough to survive independently. The brooding strategy reduces the mortality of the early life stages and ensures that the offspring are released into the environment at a size that is large enough for independent feeding. The investigation of the larval dispersal of hadal organisms reveals the mechanisms by which the organisms colonize the trench and maintain their populations in the face of the mortality that reduces the adult population. The larvae of the hadal snailfish and the juveniles of the hadal amphipods are released into the trench water, where they are carried by the currents that flow through the trench axis. The currents provide a mechanism for the dispersal of the larvae along the length of the trench, allowing the organisms to colonize new areas within the trench and to maintain genetic connectivity among the populations in different parts of the trench. The dispersal along the trench axis, which connects populations within a single trench, is fundamentally different from the dispersal between trenches, which requires the organisms to cross the abyssal barrier. The within-trench dispersal maintains the genetic connectivity of the population within a single trench, preventing the divergence of the populations at different depths and at different locations within the trench. The between-trench dispersal, which occurs rarely and on geological timescales, provides the colonization of new trenches and the occasional gene flow between trench populations, but it is too infrequent to prevent the genetic divergence that produces the endemic species in each trench. The investigation of the deep-sea lander technology that has transformed the study of the hadal zone deserves expanded treatment, because the technology has been the primary tool for the discovery of the strange biology. The hadal landers are autonomous instruments that are designed to survive the pressures of the hadal zone and to return to the surface with the data and the specimens they have collected. The landers typically consist of a frame that holds the instruments, a ballast weight that sinks the lander to the bottom, and a release mechanism that drops the ballast and allows the lander to float to the surface. The instruments carried by the landers include bait cameras that record high-definition video of the organisms attracted to the bait, baited traps that collect live specimens at hadal pressure, water samplers that collect water at specific depths for chemical and molecular analysis, and sediment corers that collect samples of the trench sediment for geochemical and microbiological analysis. The landers can be deployed for periods of hours to days, providing extended observation of the hadal community that is not possible with crude submersibles, which can remain at hadal depths for only a few hours. The development of pressurized recovery systems that maintain the specimens at hadal pressure during the ascent to the surface is one of the most important technical advances in hadal biology, because the depressurization that occurs during the normal recovery process kills the organisms and alters their molecular properties. The pressurized recovery systems, which maintain the pressure at the collection depth throughout the ascent, allow the specimens to be delivered to the surface alive and in their natural physiological state, providing specimens that can be studied under conditions that approximate the natural hadal environment. The pressurized recovery systems have been used to collect live piezophilic bacteria from hadal depths, bacteria that can then be grown in the laboratory in pressure vessels that maintain the hadal pressure. The laboratory cultivation of the piezophilic bacteria has provided the first detailed physiological characterization of organisms that require extreme pressure for growth, revealing metabolic rates, growth rates, and enzyme activities that are adapted to the conditions of the hadal zone and that cannot be replicated at surface pressure. The investigation of the community structure of the hadal zone as a function of depth within a single trench has revealed zonation patterns that divide the hadal community into depth bands, each supporting a distinct assemblage of organisms. The upper hadal zone, from roughly 6,000 to 7,000 meters, supports a community that includes both abyssal and hadal species. a transitional community that bridges the two zones. The middle Hadal zone, from roughly 7,000 to 9,000 meters, supports a community that is dominated by Hadal-specific species, including the Hadal snailfish, the large amphipods, and the Hadal holothurians. The lower Hadal zone, below roughly 9,000 meters and extending to the maximum depth, supports a community that is the most extremely adapted, dominated by the amphipods that have replaced the fish, and by the piezophilic bacteria that require the most extreme pressures. The zonation within the trench mirrors the zonation of the broader ocean, in which the surface, the mesopelagic, the bathypelagic, and the abyssal zone support distinct communities adapted to the specific conditions of each depth. The Hadal zonation adds an additional layer of depth-dependent community structure to the deep ocean, demonstrating that the biological organization of the ocean extends to the very bottom of the deepest trenches, and that the organisms at each depth are as specifically adapted to their conditions as the organisms at any other depth in the ocean. The investigation of whether the Hadal organisms are evolving faster or slower than organisms at shallower depths provides insight into the pace of evolution under extreme conditions. The prediction from the cold temperature and the slow metabolic rate of the Hadal organisms would be that the evolution is slower because the generation time is longer and the mutation rate is lower. The prediction from the high selective pressure and the extreme conditions would be that the evolution is faster because the strong selection favors rapid adaptation to the extreme environment. The molecular data suggest that the rate of molecular evolution in the Hadal organisms is comparable to, or slightly slower than the rate in related organisms at shallower depths, consistent with the prediction from the cold temperature and the slow metabolic rate. But the data are limited and the conclusion is preliminary. The philosophical dimensions of the Hadal biology deserve reflection. The Hadal organisms are the most extremely adapted organisms on the surface of the Earth, living at pressures that compress every molecule in their bodies, in darkness that has no beginning and no end, in isolation from the rest of the biosphere that is enforced by the pressure barrier of the abyssal plane. The organisms are the products of millions of years of evolution in the most extreme conditions on the planet, and their biology, when examined at the molecular level, reveals the full range of solutions that living systems can produce in response to the most demanding physical conditions. The Hadal organisms are also the most vulnerable. Their isolation means that they cannot be replaced by immigration from other habitats. Their endemism means that the loss of a trench community is the loss of species that are found nowhere else. Their slow reproduction means that the recovery from disturbance takes longer than the recovery of organisms at shallower depths. And their molecular adaptations, which are precisely selected to the specific conditions of the Hadal zone, mean that any change in those conditions, whether driven by climate change, by pollution, or by mining, could affect the viability of the organisms in ways that are difficult to predict. The Hadal zone is the most extreme, the most isolated, and the most biologically distinctive habitat on the planet. Its organisms are the most precisely adapted. Its communities are the most endemic. Its food supply is the most paradoxically enriched, and its biology, when examined in molecular detail, is the strangest and the most compelling in the natural world. The strange biology of Hadal zone creatures is not merely strange, it is revelatory. It reveals the full range of what biological molecules can achieve under extreme pressure. It reveals the precision with which natural selection can calibrate the chemistry of an organism to the physics of its environment. It reveals the capacity of isolated communities to produce endemic species through millions of years of independent evolution. And it reveals the fundamental limits of biological adaptation, the depth at which the molecular chemistry of the fish reaches its osmotic limit and the fish gives way to the amphipod, the depth at which the biology of the surface no longer applies, and the biology of the Hadal begins. The Hadal zone is the last frontier of biology on this planet. Its strange organisms are the frontier's most extraordinary inhabitants, and the biology that allows them to exist, precise and comprehensive and adapted beyond any surface expectation, is one of the most remarkable demonstrations of the capacity of living systems to persist at the absolute limits of what life can endure. The strange biology of the Hadal is not an anomaly. It is the natural consequence of extreme conditions acting on the evolutionary process over millions of years. The conditions are extreme, the evolution is precise, the organisms are extraordinary, and the Hadal zone, dark and deep and pressurized and isolated, is the place on the planet where the most extreme conditions produce the most extraordinary biology. The deepest trenches, the strangest biology, the most precise adaptations, the most extreme limits. The Hadal zone is all of these, and the organisms that inhabit it are the living proof that life, pushed to its absolute limits, finds solutions that are as extraordinary as the conditions that demand them. The specific trench comparison of Hadal communities provides biogeographic perspective that illuminates the patterns of diversity and endemism across the global Hadal zone. The Mariana Trench, the deepest trench on the planet, with a maximum depth of approximately 10,935 meters, has been the most studied Hadal habitat and the site of the first human descents. By the Trieste in 1960, and by the Deep Sea Challenger in 2012, and by the Limiting Factor in 2019. The Mariana Trench community includes the Hadal snailfish Pseudoliparaswiri, the amphipods Hirondelia gigas and several other species, xenophyrophores, polychaetes, and a diverse microbial community. The community is distinct from the communities in other Pacific trenches, with endemic species that are found only in the Mariana. The Kermadec Trench, located north of New Zealand, with a maximum depth of approximately 10,047 meters, has been studied by Hadal landers deployed by the University of Aberdeen and the University of Western Australia. The Kermadec community includes its own species of Hadal snailfish, distinct from the Mariana species, and its own assemblage of amphipods, holothurians, and microorganisms. The Kermadec and Mariana trenches are separated by thousands of kilometers of abyssal plain, and the genetic analysis of the amphipod populations in the two trenches confirms that the populations have been evolving independently for millions of years. The Tonga Trench, adjacent to the Kermadec Trench, but separated by a ridge, has a maximum depth of approximately 10,820 meters and supports a community that shares some species with the Kermadec, but that also contains endemic species not found in the Kermadec or any other trench. The proximity of the Tonga and the Kermadec trenches, separated by a relatively shallow barrier, allows some dispersal between the two, producing a pattern of shared and unique species that reflects the partial isolation of the two systems. The Japan Trench, with a maximum depth of approximately 8,020 meters, is located in one of the most seismically active regions on the planet. The Japan Trench community is affected by the frequent earthquakes that deliver turbidity currents to the trench axis, producing episodic food pulses that sustain a community that is more variable in abundance and composition than the communities in less seismically active trenches. The 2011 Tohoku earthquake, one of the most powerful earthquakes in recorded history, delivered enormous quantities of terrestrial material to the Japan Trench, and the subsequent surveys of the trench community documented changes in the abundance and the composition of the organisms that were attributed to the earthquake-driven food input. The Peru-Chile Trench, also known as the Atacama Trench, with a maximum depth of approximately 8,065 meters, is located along the western coast of South America, in one of the most productive ocean regions on the planet. The high surface productivity of the Humboldt Current system produces a food supply to the trench that is higher than the food supply in the less productive regions of the Pacific, and the Peru-Chile Trench community reflects this enrichment with higher biomass densities and a community composition that includes species that are adapted to the relatively food-rich conditions. The comparison across these trenches reveals that the Hadal communities are not uniform but are shaped by the specific conditions of each trench, including the depth, the food supply, the seismic activity, and the geographic isolation. The variation among trenches is as significant as the variation between the Hadal zone and the abyss, demonstrating that the Hadal zone is not a single habitat, but a collection of distinct habitats, each producing its own community through the interaction of the local conditions with the evolutionary potential of the organisms. The investigation of the potential commercial applications of the Hadal organism's molecular adaptations provides a practical perspective that connects the basic science to the applied. The TMAO and other osmolytes that stabilize proteins under pressure have potential applications in the pharmaceutical industry, where the stability of protein-based drugs during storage and transport is a significant concern. The protein-stabilizing properties of TMAO could be used to develop new formulations for protein drugs that are more stable and that have longer shelf lives than the current formulations. The pressure-stable enzymes of the piezophilic bacteria have potential applications in industrial biotechnology, where high-pressure processing is used in the food industry, the pharmaceutical industry, and the chemical industry. The conventional enzymes used in these processes are often inactivated by the pressures involved, requiring the addition of fresh enzyme at each step of the process. The pressure-stable enzymes of the piezophilic bacteria, which maintain their activity at the pressures used in industrial processing, could replace the conventional enzymes and reduce the cost and the complexity of the high-pressure processes. The unique lipids of the Hadal organisms, which maintain their fluidity at extreme pressures, have potential applications in the development of liposomal drug delivery systems, which use lipid vesicles to encapsulate and deliver drugs to specific tissues in the body. The lipids of the Hadal organisms, which are designed by evolution to maintain their structure and their function under extreme physical conditions, could provide the basis for liposomal formulations that are more stable and more effective than the formulations based on conventional lipids. The educational value of the Hadal zone biology lies in its capacity to illustrate the fundamental principles of adaptation, evolution, and the limits of life in a context that is both extreme and accessible. The Hadal zone organisms, with their strange adaptations and their extreme habitats, provide compelling examples of how natural selection shapes organisms at the molecular level, how geographic isolation produces endemic species, and how the physical conditions of the environment set the limits on what biology can achieve. The stories of the Hadal organisms, from the snailfish at 8,000 meters to the amphipods at 10,000 meters to the piezophilic bacteria that require extreme pressure to grow, are narratives that engage the imagination and that provide the entry point to a deeper understanding of the principles that govern life on Earth and potentially on other worlds. The Hadal zone is the deepest, the most extreme, and the most isolated habitat on the surface of the Earth. Its biology is the strangest. Its organisms are the most precisely adapted. Its communities are the most endemic. Its food supply is the most paradoxically enriched. And its significance as a standing of life's limits for the search for extraterrestrial life, for the development of new biotechnologies, and for the conservation of the planet's most extreme biodiversity, is as profound as the depth at which its organisms live. The strange biology of Hadal zone creatures is not merely a curiosity of the deep ocean, it is a window into the fundamental capabilities of living systems, a demonstration of what biology can achieve at the absolute limits of pressure, temperature, isolation, and food supply. The window reveals organisms whose molecular machinery has been remodeled by natural selection for conditions that are unique on the planet, organisms whose genomes have been rewritten, whose metabolisms have been recalibrated, and whose ecologies have been restructured for the specific demands of life at the bottom of the deepest trenches. The organisms are there. They have been there for millions of years. They are adapted, they are thriving, and they are demonstrating, with every molecule and every cell, that the limits of life on this planet are set not by the depth of the water, but by the molecular properties of the organisms themselves. The limits are further than anyone imagined before the landers descended, and the Hadal zone, dark and deep and pressurized and isolated, continues to reveal new organisms and new adaptations with every expedition that reaches its depths. The Hadal zone's strange biology is the deep ocean's most extreme expression of what life can do. The organisms are extraordinary, the adaptations are precise, the communities are unique, and the science that studies them is revealing one molecule at a time, one gene at a time, one species at a time, the full range of what living systems can achieve at the molecular limits of biological possibility. The deepest trenches in the ocean contain the strangest organisms on the planet. The strangest organisms reveal the deepest truths about life, and the deepest truths about adaptation, about limits, about the capacity of living systems to persist at the extreme, are truths that apply not only to the Hadal zone, but to every environment where life encounters the physical constraints that would limit its existence. The Hadal zone is the limit. The organisms are the proof that the limit can be reached, and the biology, strange and precise and adapted beyond any surface expectation, is the testimony of the evolutionary process working at the boundary between the possible and the impossible in the deepest, darkest, most pressurized habitat on the surface of the Earth. The strange biology of Hadal zone creatures, the deepest truth about life on this planet, written in molecules, encoded in genomes, expressed in organisms, and demonstrated every day at the bottom of the deepest trenches in the ocean, in the darkness that covers the most extreme habitat on the surface of the world. The investigation of the specific adaptations of the Hadal amphipod exoskeleton reveals molecular modifications that complement the internal biochemical adaptations and that provide additional protection against the extreme pressures. The exoskeleton of the Hadal amphipods is composed of chitin, the same structural polymer that forms the exoskeletons of all crustaceans, but the chitin in the Hadal amphipods is organized in a way that increases its resistance to compression and that provides the structural rigidity needed to maintain the body shape at pressures exceeding 1,000 atmospheres. The organization includes increased cross-linking between the chitin fibers and a higher mineral content than the exoskeletons of amphipods at shallower depths. The exoskeletal adaptations of the Hadal amphipods are complementary to the internal biochemical adaptations, providing a second line of defense against the compressive effects of pressure. The external skeleton resists the bulk compression of the body, while the internal TMAO analogues and unsaturated lipids maintain the function of the molecular machinery within the cells. The two-level defense, external structural and internal biochemical, is the comprehensive response of the amphipod body plan to the extreme pressures of the Hadal zone, and is the reason that amphipods, rather than fish, are the dominant macrofauna at the greatest Hadal depths. The investigation of the sensory biology of Hadal organisms reveals that the organisms at the deepest depths have functional sensory systems that are adapted to the specific conditions of the Hadal zone. The Hadal snailfish possesses small but functional eyes that may be used for detecting the faint bioluminescence of other organisms. The Hadal amphipods possess chemoreceptive antennae that are sensitive to the chemical traces of food in the water, allowing them to detect food falls from distances of hundreds of meters. The sensory systems are reduced compared to those of organisms at shallower depths, reflecting the diminished role of vision in the total darkness, but they are not absent, indicating that the Hadal organisms maintain the sensory capabilities that are needed for finding food, avoiding predators, and locating mates in the extreme conditions. The investigation of the acoustic environment of the Hadal zone reveals that the trenches have distinct acoustic properties that affect the propagation of sound within the trench. The V-shaped geometry of the trench creates an acoustic waveguide that channels sound along the trench axis, potentially allowing organisms at one end of the trench to detect sounds produced at the other end. The acoustic waveguide effect may provide the Hadal organisms with long-range communication and detection capabilities that are not available on the flat abyssal plain, where sound propagates freely in all directions. The sounds produced by the Hadal organisms, including the clicks and rasps recorded by acoustic sensors deployed on Hadal landers, indicate that the Hadal community is acoustically active, with organisms producing sounds that may serve functions in communication, in predator detection, and in territory defense. The acoustic activity of the Hadal community is consistent with the overall picture of the Hadal zone as a biologically active habitat, not the silent, lifeless zone that was once assumed. The investigation of the deep-sea trenches as analogues for extraterrestrial environments has identified the Hadal Zone as the most relevant terrestrial analogue for the high-pressure environments that are expected to exist in the interiors of icy moons and on the ocean floors of water worlds. The Hadal organisms, which function at pressures of 600 to more than 1,000 atmospheres, demonstrate that life can operate at pressures that are comparable to the pressures expected at the bottom of Europa's ocean, estimated at roughly 300 to 400 atmospheres, or at the bottom of Enceladus's ocean, estimated at roughly 100 atmospheres. The Hadal organisms demonstrate that the pressure barrier is not a barrier to life, that biological molecules can be adapted to function at extreme pressures, and that the energy sources available from rock-water interactions in the Hadal zone, including the hydrogen from serpentinization and the organic matter from the food funnel, may be analogous to the energy sources available in extraterrestrial oceans. The NASA Ocean Worlds program, which is developing mission concepts for the exploration of the oceans of Europa and Enceladus, has identified the Hadal Zone as a priority study site for the development of life detection technologies. The instruments that would be needed to detect life in an extraterrestrial ocean, including molecular sensors, pressure-tolerant cameras, and sample return systems, are being tested in the Hadal Zone, where the conditions provide the closest terrestrial approximation to the conditions expected in the target environments. The connection between the Hadal biology and the concept of the habitable zone extends the significance of the Hadal Zone beyond the Earth. The traditional habitable zone, the region around a star where surface conditions allow liquid water, is the primary criterion for identifying potentially habitable planets. The Hadal organisms demonstrate that life does not require surface conditions, that life can exist in high-pressure, dark, isolated environments that are sustained by the internal energy of the planetary body, rather than by the energy of the star. The demonstration expands the concept of habitability from the surface to the subsurface, from the star-dependent to the planet-dependent, and from the narrow traditional habitable zone to the much broader range of environments where liquid water and chemical energy coexist. The investigation of the evolutionary origins of the Hadal fauna reveals that the Hadal organisms are descended from shallower water ancestors that colonized the trenches as the trenches formed, adapting progressively to the increasing depth and the increasing pressure as the subduction process deepened the trench over geological timescales. The colonization was not a single event, but a continuous process, with organisms entering the trench from the surrounding abyssal plain and from the surface, and gradually evolving the adaptations needed for life at progressively greater depths. The evolutionary trajectory from abyssal to Hadal involved the progressive increase of the TMAO concentration, the progressive modification of the lipid composition, the progressive loss of unnecessary genes, and the progressive amplification of the genes needed for Hadal life. The trajectory is not complete. The Hadal organisms are still evolving, still adapting to the conditions of the trenches, still producing new species through the independent evolution of isolated populations. The evolutionary process in the Hadal Zone is ongoing, driven by the same mechanisms of mutation, selection, and genetic drift that drive evolution everywhere, but operating under the most extreme conditions and producing the most extreme adaptations that the evolutionary process has generated on the surface of the planet. The potential impact of ocean warming on the Hadal Zone adds urgency to the study of the Hadal biology. The warming of the surface ocean is producing a warming signal that is slowly propagating into the deep ocean, and models predict that the warming will eventually reach the Hadal Zone, increasing the temperature by fractions of a degree over periods of decades to centuries. The warming, though small in absolute terms, could affect the organisms that are adapted to the near-constant temperatures of the Hadal Zone, organisms whose molecular adaptations are calibrated to the current temperature, and that may not function correctly at even slightly elevated temperatures. The detection of the warming signal in the Hadal Zone requires long-term temperature monitoring at Hadal depths, monitoring that has been initiated at a few locations but that has not been maintained at the scale needed for the detection of small temperature trends against the natural variability. The establishment of permanent monitoring stations in the Hadal zone, equipped with temperature sensors, chemical sensors, and biological samplers, is a priority that has been identified by the scientific community, but that has not been implemented at the scale required. The investigation of the chemical pollution of the Hadal zone extends beyond the microplastics that have been documented in the trench sediment to include persistent organic pollutants, including polychlorinated biphenyls and polybrominated diphenyl ethers that have been detected in the tissues of Hadal amphipods at concentrations that exceed the concentrations in organisms from less polluted shallow water habitats. The high concentrations of pollutants in the Hadal organisms are believed to reflect the bioaccumulation of the pollutants as they are transferred from the surface to the deep through the food web, with each trophic transfer concentrating the pollutants in the tissues of the consumer. The Hadal organisms, at the end of the trophic chain, accumulate the highest concentrations. The detection of persistent organic pollutants in the Hadal organisms demonstrates that the human chemical footprint extends to the most remote habitats on the planet, and that the Hadal zone is not immune to the effects of surface pollution. The health effects of the pollutants on the Hadal organisms are unknown but are a concern, because the pollutants can disrupt the endocrine system, the immune system, and the reproductive system of the organisms that accumulate them. The Hadal amphipods, which are the dominant macrofauna at the greatest depths and which accumulate the highest concentrations of pollutants, may be the organisms most affected by the chemical pollution of the Hadal zone. The investigation of the Hadal zone is a story of discovery that is still in its earliest chapters. The first chapter described the existence of life at Hadal depths. The second chapter described the adaptations that allow life to function at extreme pressures. The third chapter described the molecular mechanisms of the adaptations. The fourth chapter described the genomic basis of the adaptations. The fifth chapter described the ecological dynamics of the Hadal communities. The chapters that remain to be written will describe the full range of Hadal biodiversity, the full range of Hadal adaptations, the full range of Hadal ecological interactions, and the response of the Hadal communities to the environmental changes that are being driven by human activity. The writing of these chapters requires sustained investment in Hadal research, investment that includes the development and deployment of Hadal landers, the training of specialists in Hadal taxonomy and molecular biology, the maintenance of long-term monitoring programs at Hadal depths, and the international coordination of research efforts across the global system of Hadal trenches. The investment is justified by the importance of the Hadal zone as the most extreme biological habitat on the planet, by the uniqueness of its organisms, by the potential applications of its molecular adaptations, and by the vulnerability of its communities to the environmental changes that are affecting the deep ocean. The strange biology of Hadal zone creatures is not a niche curiosity of deep ocean science. It is a fundamental chapter in the story of life on Earth, a chapter that reveals the absolute limits of biological adaptation, the molecular mechanisms by which life functions at extremes, and the evolutionary processes by which isolated communities produce endemic species. The chapter is still being written. The landers are still descending. The specimens are still being analyzed. And the Hadal zone, dark and deep and pressurized and isolated, is still revealing its strange organisms to the science that is only now developing the tools to find them. The deepest trenches, the strangest biology, the most extreme adaptations, the most isolated communities, the most precise molecular calibrations. The Hadal zone is all of these, and the organisms that inhabit it, living at the limits of what life can achieve, are the most extraordinary products of the evolutionary process on the surface of the Earth. The strange biology of Hadal zone creatures is the biology of life at its limits. The limits are precise, the adaptations are extraordinary, and the organisms, thriving in the darkness at the bottom of the deepest gashes in the planet's surface, are the living proof that the limits of life are further than anyone imagined, further than any model predicted, and further than any organism outside the Hadal zone has ever reached. The specific molecular biology of the Hadal Trench sediment communities provides an additional dimension of strangeness that complements the strangeness of the macrofaunal biology. The sediment at the bottom of the Hadal trenches contains microbial communities whose metabolic capabilities include the anaerobic oxidation of methane, the chemoautotrophic fixation of carbon using hydrogen as an energy source, and the reduction of metals, including iron and manganese, as electron acceptors. These metabolic processes are not unique to the Hadal zone, but they occur at the Hadal zone in the context of extreme pressure and in sediments whose organic matter content has been enriched by the food funnel effect, creating conditions that are unique to the Hadal environment. The rate of microbial activity in the Hadal sediment, measured by the uptake of labeled substrates and by the production of metabolic waste products, is higher than the rate in the sediment of the adjacent abyssal plain. Consistent with the higher organic matter content and the more active community, the higher microbial activity in the Hadal sediment contributes to the higher rates of nutrient cycling, carbon remineralization, and gas production that have been measured in the trench sediment, and it supports the conclusion that the Hadal zone is a more biologically active habitat than the abyss. The investigation of the Hadal viruses, the viruses that infect the bacteria and archaea of the Hadal sediment, has revealed a viral community that is as extreme as the microbial community it infects. The Hadal viruses include phages, viruses that infect bacteria whose morphology and genomic organization are adapted to the high-pressure conditions and whose infection dynamics are governed by the slow growth rates of the host bacteria. The viral lysis of the Hadal bacteria releases dissolved organic matter into the pore water, contributing to the nutrient cycling of the Hadal sediment through the viral shunt, the same process that has been documented in the subsea floor biosphere at shallower depths. The investigation of the Hadal meiofauna, the organisms between roughly 32 micrometers and 1 millimeter in size, has revealed abundance patterns that reflect the food funnel enrichment. The meiofaunal abundance in the Hadal sediment is higher than the abundance on the adjacent abyssal plain, with the nematodes, the most abundant group, reaching densities that are several times higher in the trench than on the plain. The higher meiofaunal abundance supports higher levels of predation by the macrofauna, including the amphipods and the polychaetes, creating a food web that is more productive and more complex than the food web on the abyssal plain. The investigation of the biochemistry of the Hadal amphipod Eurythenes gryllus, one of the most widely distributed deep-sea amphipods, has revealed molecular adaptations that differ between populations from different depth ranges. The populations from Hadal depths possess higher TMAO analog concentrations, different lipid compositions, and different enzyme kinetics from the populations from abyssal depths, demonstrating that the molecular adaptations vary with depth, even within a single species. The depth-dependent variation in molecular biology within a species is consistent with the concept that the Hadal zone imposes specific molecular requirements that differ from the requirements at abyssal depths and that require specific molecular solutions. The investigation of the reproductive timing of Hadal organisms has revealed seasonal patterns that are surprising for organisms at depths, where the environmental conditions are believed to be constant throughout the year. The seasonal patterns, documented by the variation in the proportion of reproductive females in the amphipod populations sampled at different times of the year, suggest that the Hadal organisms can detect and respond to seasonal changes in the food supply that are driven by the seasonal variation in surface productivity, and that are transmitted to the Hadal zone through the marine snowflux. The ability to detect seasonal signals at Hadal depths indicates that the organisms are more responsive to environmental changes than the constant physical conditions of the Hadal zone would suggest. The connection between the Hadal biology and the broader question of how life responds to extreme conditions provides the most important scientific context for the study of the Hadal organisms. The Hadal organisms are the most extremely adapted organisms on the planet, and the study of their adaptations provides insights into the fundamental mechanisms by which living systems can be modified by natural selection to function under conditions that are at the absolute limits of biological possibility. The insights are relevant not only to the understanding of life on Earth, but also to the understanding of life's potential elsewhere in the universe, on the ocean worlds of the outer solar system, and on the rocky planets of other stars, where the conditions may include pressures, temperatures, and chemical environments that are comparable to those in the Hadal Zone. The Hadal Zone biology is therefore not a narrow speciality of deep ocean science. It is a fundamental branch of biology that addresses the most extreme capabilities of living systems, and that provides the data needed for the assessment of life's potential in the most extreme environments. The study of the Hadal organisms is the study of life at its limits, and the limits, as the organisms demonstrate, are further than anyone imagined before the landers descended and the molecules were measured. The final dimension of the Hadal Zone biology that deserves reflection is the sheer improbability of the organisms that inhabit it. The Hadal Zone is the most extreme habitat on the planet. The organisms that inhabit it have been adapted to the extreme conditions by millions of years of natural selection. The adaptations are precise, comprehensive, and molecular. The organisms are thriving, and the biology that allows them to thrive is as strange, as specific, and as extraordinary as any biology on the planet. The Hadal organisms are the evolutionary products of the most demanding conditions that the surface of the Earth has to offer. Their existence demonstrates that life can function at pressures exceeding 1,000 atmospheres, that biological molecules can be precisely calibrated to the physics of the environment, that isolated communities can produce endemic species through millions of years of independent evolution, and that the limits of life are not set by the depth of the water, but by the molecular properties of the organisms themselves. The Hadal Zone is the planet's most extreme biological laboratory. Its organisms are its most extreme products, and the strange biology that defines them is the most extreme demonstration of what the evolutionary process can achieve when the conditions are extreme and the time is sufficient. The strange biology of the Hadal Zone is not an anomaly. It is a natural consequence of extreme conditions acting on the evolutionary process over geological timescales. The result is extraordinary. The organisms are precise, the adaptations are comprehensive, and the Hadal Zone, dark and deep and pressurized and isolated, is the place on the planet where the most extraordinary biology is produced, tested, and refined by the most demanding conditions that the surface of the Earth can provide. The investigation of the Hadal organisms' capacity for phenotypic plasticity, the ability to modify their biology in response to changing conditions without genetic change, reveals an additional dimension of adaptability that complements the genetically encoded adaptations. The phenotypic plasticity of the Hadal organisms includes the ability to modify the lipid composition of their membranes in response to changes in temperature and pressure, the ability to adjust the concentration of osmolytes in response to changes in the salinity of the pore water, and the ability to modify the expression of specific genes in response to changes in the food supply. The plasticity provides a short-term response mechanism that allows the organisms to accommodate environmental variation that is too rapid for genetic adaptation, including the variation in food supply that is driven by the episodic earthquake-triggered turbidity currents and by the seasonal variation in the marine snow flux. The phenotypic plasticity of the Hadal organisms is constrained by the same molecular limits that constrain the genetically encoded adaptations. The organisms can modify their lipid composition within the range that is compatible with membrane function at Hadal pressures, but they cannot modify it beyond this range. The organisms can adjust their osmolyte concentrations within the range that is compatible with protein function and osmotic balance, but they cannot adjust them beyond this range. The plasticity provides flexibility within the limits, but it does not extend the limits themselves, which are set by the fundamental properties of the biological molecules. The investigation of the potential for synthetic biology applications based on the Hadal adaptations provides a forward-looking perspective that connects the basic science to the applied. the genes that encode the pressure-stable proteins, the unsaturated lipid-producing enzymes, and the TMAO synthesizing pathways of the hadal organisms could be transferred to other organisms using genetic engineering techniques, conferring pressure tolerance on organisms that do not naturally possess it. The engineered pressure-tolerant organisms could be used in industrial biotechnology, where high-pressure processing is used in food sterilization, in chemical synthesis, and in the production of pharmaceuticals. The transfer of hadal genes to industrial organisms is technically feasible but has not been widely pursued because the applications of pressure-tolerant biotechnology are currently limited and because the expression of the hadal genes in a non-hadal host requires the co-expression of multiple genes that work together to produce the complete pressure tolerance phenotype. The complexity of the pressure tolerance system, which involves modifications to proteins, membranes, and metabolic pathways simultaneously, makes the genetic engineering of pressure tolerance more difficult than the engineering of single gene traits and requires a systems biology approach that considers the interactions among all the components of the pressure tolerance system. The future of hadal biology depends on the continued development and deployment of the technologies that provide access to the hadal zone, the landers, the submersibles, the autonomous vehicles, and the pressurized recovery systems that collect specimens at hadal pressure. The technologies are improving rapidly, driven by advances in materials science, in battery technology, in sensor miniaturization, and in autonomous navigation. The improving technologies are providing access to the hadal zone that was not possible even a decade ago, and the rate of discovery is accelerating as a result. The rate of discovery in the hadal zone is currently limited not by the technology but by the funding that is needed to build the instruments, to deploy them at hadal depths, to recover and analyze the specimens, and to train the specialists who can interpret the results. The funding for hadal research is provided by national science agencies, by international research programs, and by private foundations, and the total investment is modest, perhaps tens of millions of dollars per year across all nations and all programs. The investment is sufficient for the current rate of discovery, but is not sufficient for the comprehensive characterization of the hadal zone that its importance warrants. The hadal zone is one of the most important habitats on the planet for the understanding of life's limits, for the conservation of unique biodiversity, and for the development of biotechnologies based on extreme adaptations. The investment in its study is a small fraction of the investment in other scientific fields that address comparably fundamental questions. The expansion of the investment to a level that matches the importance of the habitat would accelerate the rate of discovery, would provide the baseline data needed for the protection of the hadal communities, and would reveal the full range of strange biology that the hadal zone contains. The hadal zone has been producing its strange organisms for millions of years. The science has been studying them for decades. The discoveries have been extraordinary, revealing molecular adaptations that no one predicted, genomic modifications that no one anticipated, ecological dynamics that no one expected, and organisms whose biology is as strange and as beautiful as any on the planet. The discoveries continue. The hadal zone continues to reveal new organisms and new adaptations with every lander that descends and every specimen that is analyzed. The strange biology of hadal zone creatures is the deep ocean's most extreme and most compelling story. The story is being written by the organisms themselves, in molecules and genes and adaptations, in the darkness and the pressure of the deepest trenches on the planet. The science is reading the story, one specimen at a time, one gene at a time, one adaptation at a time, and the reading, far from being complete, has barely begun. The hadal zone's organisms are extraordinary. Their biology is strange. Their adaptations are precise. Their communities are unique. And the science that studies them is revealing, decade by decade, the full range of what life can achieve at the absolute limits of biological possibility, in the deepest, darkest, most pressurized, and most isolated habitat on the surface of the Earth. The strange biology of hadal zone creatures, the deepest life, the strangest molecules, the most precise adaptations, the most extreme limits, and the most compelling evidence that life, pushed to its absolute boundary, finds solutions that are as extraordinary as the conditions that demand them. The hadal zone is the edge, the organisms are the proof, and the proof, living and thriving and adapted in the darkness at the bottom of the deepest trenches, is one of the most remarkable facts about life on this planet. The investigation of the relationship between trench geometry and community structure has revealed that the shape of the trench, its width, its depth, its slope angle, and its curvature affects the food funnel efficiency and therefore the food supply to the community. Narrow trenches concentrate the food more effectively than wide trenches, producing higher food supply per unit area at the trench axis. Deep trenches receive food from a larger catchment area of the surrounding abyssal plain than shallow trenches, producing a total food input that is proportional to the depth. The variation in trench geometry across the global system of hadal trenches creates a range of food supply conditions that support communities of different sizes, different structures, and different levels of activity. The investigation of the role of the hadal zone in the geological carbon loss cycle reveals that the trenches may be significant sinks for organic carbon on geological timescales. The carbon that is delivered to the trench axis by the food funnel and by the earthquake-driven turbidity currents is buried in the rapidly accumulating sediment of the trench and is transported into the Earth's interior by the subduction process that created the trench. The carbon that enters the subduction zone is removed from the ocean atmosphere system for geological timescales of millions to hundreds of millions of years, making the hadal trenches potentially significant long-term carbon sinks, whose contribution to the global carbon budget has not been fully quantified. The quantification of the carbon sink function of the hadal trenches requires the measurement of the carbon input rate, the carbon burial rate, and the subduction rate at each of the global system of trenches, measurements that have been made at only a few locations and that are far from sufficient for a global estimate. The preliminary estimates suggest that the total carbon burial in the hadal trenches may be significant when summed across all 33 trenches worldwide, but the uncertainty is large and the estimates should be regarded as preliminary. The connection between the hadal biology and the origin of life hypothesis provides a speculative but scientifically grounded perspective on the significance of the hadal organisms. If life originated in high-pressure environments, as some origin of life hypotheses propose, the hadal organisms may represent the descendants of the earliest life forms, organisms whose molecular adaptations for high pressure are not derived innovations, but are ancestral features that have been retained from the earliest period of life's history. The hypothesis is speculative and is not supported by definitive evidence, but it is consistent with the phylogenetic placement of some piezophilic organisms near the base of the tree of life, and with the chemical conditions in the hadal zone that are compatible with some models of prebiotic chemistry. The investigation of the hadal zone continues to produce discoveries that challenge the existing understanding and that expand the known range of biological adaptation. Each expedition that deploys landers to the hadal zone recovers specimens that include organisms that have not been described, that possess adaptations that have not been documented, and that reveal ecological relationships that have not been anticipated. The rate of discovery shows no sign of slowing, and the hadal zone remains one of the most productive frontiers of biological exploration on the planet. The strange biology of hadal zone creatures is not a solved problem. It is an open question, a question that is being addressed by the ongoing research, but that is far from answered. The molecular adaptations are documented, but not fully understood at the mechanistic level. The genomic modifications are sequenced, but not fully characterized at the functional level. The ecological dynamics are observed, but not fully modeled at the quantitative level. And the evolutionary processes that produce the endemic species are inferred, but not fully documented at the genetic level. The open questions provide the agenda for the next generation of hadal research, an agenda that includes the mechanistic understanding of the pressure adaptations, the functional characterization of the genomic modifications, the quantitative modeling of the ecological dynamics, the genetic documentation of the evolutionary processes, and the assessment of the vulnerability of the hadal communities to the environmental changes that are being driven by human activity. The agenda is ambitious, the tools are available, the organisms are waiting in the trenches, and the discoveries that the agenda will produce will further expand the understanding of life at its limits and will further demonstrate the extraordinary capacity of living systems to adapt to the most extreme conditions on the surface of the Earth. The Hadal Zone creatures are strange because the Hadal Zone is extreme. The extremity produces the strangeness reveals the limits, and the limits, as the organisms demonstrate with every molecule and every cell, are further than anyone imagined and further than any model predicted. The strange biology of Hadal Zone creatures is the planet's most extreme lesson in what life can do. The lesson is taught in molecules and genes and organisms, in the darkness and the pressure of the deepest trenches, by the strangest and most precisely adapted organisms that the evolutionary process has ever produced on the surface of the Earth. The lesson is extraordinary, the organisms are extraordinary, and the Hadal Zone, which produces them both, is the most extraordinary biological habitat on the planet. The deepest life, the strangest biology, the most extreme adaptations. The Hadal Zone contains all of these, and the science that studies them continues to reveal, one discovery at a time, the full and extraordinary range of what life can achieve at the absolute limits of biological possibility. The strange biology of Hadal Zone creatures stands as the final, most extreme chapter in the story of life's adaptation to the deep ocean. The chapter builds on every previous chapter, on the adaptations of the mesopelagic, the bathypelagic, and the abyssal organisms, and extends them to the ultimate extreme, the pressures exceeding 1,000 atmospheres, the isolation enforced by the abyssal barrier, the food enrichment of the funnel-shaped trench, and the molecular modifications that allow proteins to fold, membranes to flow, enzymes to catalyze, and genomes to replicate at conditions that would disable every surface organism. The chapter is not finished. The Hadal Zone continues to produce new discoveries with every expedition. The molecular biology of the Hadal organisms continues to reveal new adaptations with every genome sequenced and every protein analyzed. The community ecology of the Hadal trenches continues to reveal new patterns of endemism, new food web relationships, and new connections between the Hadal and the surface through the food funnel and the marine snow flux. The story is ongoing, written by the organisms in their molecules and expressed through their strange, beautiful, precisely adapted bodies in the darkness at the bottom of the deepest trenches on the planet. The Hadal Zone is the planet's deepest habitat. Its organisms are the planet's most extremely adapted. Its biology is the planet's strangest. And the science that studies it, which has been operating for only a few decades with the lander technology that provides access to the extreme depths, is revealing the full and extraordinary range of what life can achieve at the molecular limits of biological possibility, one specimen at a time, one molecule at a time, one adaptation at a time. The organisms of the Hadal Zone are the deep ocean's final argument for the breadth of biological possibility. They are the living proof that life can function at the most extreme pressures on the surface of the Earth, through molecular modifications that are as precise as they are comprehensive. They are the evidence that geographic isolation produces endemic species, that extreme conditions produce extreme adaptations, and that the evolutionary process, given sufficient time and sufficient pressure, produces organisms whose biology is as extraordinary as the conditions that shaped them. The Hadal Zone is the edge of life on this planet. Its organisms have reached the edge, and the biology they possess, documented in the laboratories of the surface and demonstrated in the darkness of the deep, is one of the most remarkable collections of molecular adaptations that the natural world has produced. Adaptations that reveal the full range of what living systems can achieve when the conditions are extreme and the evolutionary time is sufficient. The strange biology of Hadal Zone creatures is the deep ocean's most profound message about the nature of life. The message is that life adapts, life persists, life finds solutions to problems that seem unsolvable, life functions at extremes that seem impossible, and life, at the bottom of the deepest trenches in the ocean, in the darkness, and the pressure and the isolation, thrives with a precision and an elegance that reveals the full and extraordinary capacity of the evolutionary process. The Hadal zone's organisms are strange, their biology is extreme, their adaptations are precise, and their existence, at the bottom of the deepest gashes in the surface of the Earth, is one of the most remarkable facts about life on this planet. The deepest trenches, the strangest biology, the most extreme life. The Hadal zone is where the limits of life are drawn, and the organisms that live there demonstrate every day that the limits are further than anyone knew. The Hadal zone will continue to reveal its strange organisms for as long as the science has the instruments to reach them, the specialists to study them, and the funding to sustain the research. The organisms are there, in every trench, at every depth from 6,000 to nearly 11,000 meters, adapted to conditions that are found nowhere else on the planet, possessing biology that is described nowhere else in the scientific literature, and living their extraordinary lives in the most extreme habitat that the surface of the Earth provides. The biology of the Hadal organisms is strange because the conditions are extreme. The conditions are extreme because the trenches are deep. The trenches are deep because the tectonic plates converge, and one dives beneath the other, creating the gashes in the seafloor that provide the habitat for the most extremely adapted organisms on the planet. The geology creates the habitat creates the conditions create the adaptations, and the adaptations, strange and precise and molecular, create the organisms that inhabit the deepest, most pressurized, most isolated, and most biologically distinctive habitat on the surface of the Earth. The strange biology of Hadal zone creatures is the product of this chain, from geology to habitat, to conditions to adaptations to organisms, a chain that connects the movement of the tectonic plates to the molecular properties of the proteins in the cells of the organisms that live at the bottom of the trenches that the plates created. The chain is as long as the history of the Earth and as deep as the deepest trench, and the organisms at the end of the chain, the strange, precisely adapted creatures of the Hadal zone, are as extraordinary as any organisms that the planet has produced. The Hadal zone is the limit. The biology is strange, the organisms are extraordinary, and the science, still in its earliest stages, continues to reveal the full and remarkable range of what life can achieve at the absolute extreme of biological possibility, in the deepest darkness, under the greatest pressure, in the most isolated habitat, on the surface of the planet we call home. The Hadal zone is the edge of life. The organisms that live there are the proof that the edge can be reached, inhabited, and transformed into a home. End of strange biology of the Hadal zone is the deep ocean's most profound and most extraordinary lesson about the capacity of life to adapt, to persist, and to thrive at the absolute boundary between the possible and the impossible. The strange biology of the Hadal zone is the deep ocean's most profound and most extraordinary lesson about the capacity of life to adapt, to persist, and to thrive at the absolute boundary between the possible and the impossible. The Hadal zone is the edge of life. The organisms that live there are the proof that the edge can be reached, inhabited, and transformed into a home.